no worries, no worries. All right. So the next thing that we're going to talk about is the complement pathway. And that will conclude our journey through the immune system. So one thing to remember is I've chunked all the lectures up and I've talked about things individually, and I've said, okay, here is the innate. Here is the adaptive required immune system. Here is inflammation. Here is this, here is that. One thing you got to remember is all this is all happening all at the same time. So everything is happening at the same time. And this is one more thing that's going on. And this is a non-specific system that's going on in the background. And what the complement system is it's a system of proteins within your blood. And there are a couple dozen of these proteins. There are 20 or more of them. And as you can see, we're only talking about a very few of them. So again, we are going to super simplify this so we can gain some degree of understanding of what's going on here. Okay? But what happens with the complement system is I look at it as it complements all the other immune things, all the other immune functions going on. It helps complement them. It helps that out a little bit. Okay? And so there are three general pathways. All right? There's what we call the classical pathway, the Mannose binding lectin pathway, or the MBL pathway, and then there is the alternative pathway. Okay? Now, the classical pathway is where I'm going to start, and the reason it's called the classical pathway is simply because it's the first one that we were able to find. We identified it first, so we called it the classical pathway. All right, nothing more special than that. So, what happens here is when antibodies bind to antigens on the pathogen, the classical pathway gets activated. And the main take-home point that you want to think about is it's the C3 protein that ultimately changes into the active processes that are going on. Okay, again, we said there are 20 or more of these proteins, but it's C3, and what happens to complement 3 that we're really concerned about, right? So we're kind of cutting through a lot of the red tape, if you will. Okay, so what happens is complement 1, C1, actually binds to the antigen. All right, so let me draw my microorganism here. Okay, so there's my microorganism, and let's say that microorganism has some antigen on its cell wall, and what happens is complement 1, okay, binds. All right, so there's complement 1, C1 binds to that antigen, and that allows for C3 to come in and bind. All right, and when that happens, that forms an enzyme known as C3 convertase. C3 convertase is also known as C4B2A complex. Oh my God, the names, right? All right, so C3 convertase is formed, okay? C4B2A complex, all right? All right, so C3 down this pathway can become C3A, its activated form. And what this does is this enhances inflammation. Okay, when this particular pathway is activated, this helps with mast cell degranulation. It helps cause more mast cells to degranulate, which causes more increased capillary permeability, right? Causes more blood flow into the area, more swelling, etc. Okay? So that's what's happening down the classical pathway. Okay? You have antibodies binding to antigens. You've got C1 binding to the antigen. Okay? okay? C1 then forms C3 convertase with C3, okay? And that ultimately enhances or modulates the inflammatory response through mast cell degranulation. You guys good with that? That's a lot. That's a lot, huh? Okay. Now, the other pathway, the Mannose binding lectin pathway, is very similar to the classical pathway. Okay, it's very similar. But instead of C1, or instead of binding to an antigen, which is a protein, antigens tend to be proteins, okay, instead, okay, there are other molecules like MASP and MBL, and these molecules don't bind to proteins, don't bind to antigens, but rather they bind to sugar molecules embedded within the surface of that pathogen, all right? So remember, when you look at a cell wall, that cell wall has sugar molecules and proteins and channels and antigens and pumps and lipids and all kinds of things hanging off of it, right? In this case, the binding is to an antigen. In the Mannose binding lectin pathway, what happens is it's binding to sugar molecules on the surface of those pathogens, okay? And that forms C3 convertase as well as C3 convertase here, okay?
What is it? But similar to classic, well, but involves what?
MASP and MBL. Yeah, those are just the bonding proteins instead of the C1 here.
And is it phosphorate, the same?
MASP, MASP, and MBL, ... means others and others.
What's this one I was familiar with?
What's that? Oh, I'll talk about that here. Yeah, I'm not quite there. Are you guys here though? Are you guys okay with what the difference is between the Mannose binding lectin pathway and the classical pathway? It's antigen here in the classical and sugar molecule on the surface here. Does that make sense? Cool. All right. So, ultimately C3 can be turned to C3A, which enhances inflammation, or over here, C3B. And C3B, complement C3B, can cause one or two things to happen. It can cause something known as opsonization, okay? And so what happens is it binds to, so here's my pathogen, and C3 convertase, well, let me keep the colors, will bind, okay? And what that does is, as it binds to these pathogens, it opsonizes them. It makes them easier, okay? It makes it easier for the phagocytic cells to phagocytize them. So basically what happens is when a neutrophil or dendritic cell comes along, and the color here, all right, and it begins to phagocytize, there are receptors that line up with the C3 convertase there, that on that opsonized cell, right? And so it allows it to get a hold of that pathogen and engulf it. Does that make sense? That's what opsonization does. Opsonization allows the phagocytic cell to phagocytize that pathogen easier. Does that kind of make sense? Okay. And the other thing that can happen is this MAC can be formed. And that stands for membrane attack complex. MAC, membrane attack complex. And what membrane attack complex is basically, you can look at it as kind of like a large needle is developed, and it gets shot into the pathogen and puts a big pore, a big hole into the wall of the pathogen. It puts a big pore in there, and then what happens? It basically spills its guts out all over the place and dies. Yeah, it's pretty crazy, huh? And it's pretty crazy when you think about it, like this stuff is going on inside of our body here. So ultimately, there are three major things that occur that the complement pathway does. Is it enhances or modulates inflammation, it causes opsonization of the pathogen, or it can help produce a membrane attack complex that puts a pore through the cell wall of the pathogen. Okay, those are the three major things that we see coming out of the complement system, the complement pathway. And then the final pathway is called the alternative pathway, all right? And this is when C3 convertase catalyzes C3b production. And ultimately what this does, again, going super simplified, is it enhances the other two pathways. Okay, it enhances the classical pathway, it enhances Mannose binding Lectin pathway. Does that kind of make sense there? So that's what the complement system is it's complementary. It's complementary to all of the other immune system processes that are occurring. It's just one more layer of generally non-specific stuff going on. You guys okay with that? Yeah. Kind of makes sense there? Excellent. All right. So that's all of our immune system stuff. So let's go ahead and